Oh, here I started one, Jasmine.
Awesome. So hi everyone, thank you for coming. My name is Jasmine and I'm the founder and president of CISSA. CISSA stands for the Calgary Indigenous STEAM Students Association. And I'm really excited to see everyone here tonight. Thank you to our professors for taking the time out of your busy evenings to come and speak to us. We're really excited to hear from you. An introduction to our club is that CISSA aims to serve as a platform for Indigenous and non-Indigenous U of C students who are interested in promoting Indigenous engagement in STEAM, participating in learning opportunities to strengthen the campus community, and exploring professional development opportunities. Our organization is open to both students and non-students, as well as Indigenous and non-Indigenous members, so you're welcome to join us, and you can do so by emailing communications.cissa@gmail.com. This event will be recorded, so if you do choose to turn on your camera or submit a question, this constitutes consent for your video, image, or question to be shared. The recording of this session will be made available online in the future, so make sure you follow us on Instagram, Snapchat, Facebook, or LinkedIn to stay in the loop with what we're doing. We'll just get started with a land acknowledgement. So we want to acknowledge that our university is located on Treaty 7 territory, which is Blackfoot territory, including the Siksika, Piikani, and Kainai. And it's also home to the Tsuut'ina, the Stoney Nakoda, and the Métis Nation of Alberta, Region 3. We, as a club, work to strive to respect and incorporate these Indigenous ways of knowing and being into our respective practices and encourage others to do the same. And before we begin, we'd like to thank our Diamond Tier sponsor, TC Energy. We're thankful for their support in making this event possible. So tonight's event will begin with an introduction to research presentation given by Becca, who's our awesome VP events. And then we're going to have individual presentations from professors. If you can save your questions until the end of the event, we'll have breakout rooms and you can ask questions directly. With that, let's welcome Becca.
All right. Thank you so much for the introduction and the land acknowledgement, Jasmine. So, a little bit about me. My name is Becca. I am a fifth year kinesiology student here at the University of Calgary, as well as I have done 2 summers of undergraduate research, and I'm currently completing an honors project in biomechanics within the Faculty of Kinesiology. So, we figured with seeing as we're a STEAM kind of club and with research deadline or grant deadlines coming up for undergraduate students, we'd run this event to kind of help give undergraduate students an idea of what it's like to be involved, as well as what kind of goes on and how to get involved, reach out to supervisors, as well as apply for these grants. So the first thing that as an undergraduate I would recommend is look into the awards that you're eligible for and make note of any deadlines. If chances are good from talking with my supervisor, he is more interested in students who have an idea and are interested in applying for grants than students who aren't and don't have any idea what's going on. So the key thing here is to look into it and be proactive. So there are a lot of different awards that are available for summer research. Within the Faculty of Kinesiology, I know of 3 that are available to any kine student. So we're a pretty small faculty, so I'm sure there's so many for the other faculties as well. I will kind of send this link around with, as well as the recording after. So, or you can just go to ucalgary.ca/students under for research, and it'll have all the grants that are eligible for undergraduates. So one thing that is good to be aware of is that there's a common award application, which means that you can apply for multiple awards with one application. It's really nice. It kind of makes the time a lot easier, and it's one form that you just fill out and you check boxes for which ones you're eligible for. So the 3 kind of big ones that I'm aware of are the Peer Award, which is in the Common Apps applications for undergraduates. And the only prerequisite here, as it says, is you have to have a GPA of 3.2 or higher and be in an undergraduate degree program. So it's available to anyone who is interested in research, not just a particular faculty. The NSERC Awards are a national award, so it's a pretty prestigious one, and it's one that a lot of grad students also get. So this is an undergraduate version. as well. So it's specifically for natural sciences and engineering. So it can't unfortunately be health sciences related, but you can, if you are doing something within kind of health sciences, you just have to tailor the research specifically to be more general natural science or engineering as well. So you have to be a Canadian citizen and have a GPA of 3.0. And then Alberta Innovates is the award that I have received twice. So there are a number, so it's one of the many awards in the common undergraduate application as well. And these are open to students pursuing specifically health related research, and you have to have a GPA of 3.2 or higher. So then the next step, once you have an idea of the awards that you're eligible for, is you want to find a professor that you want to work with. This can be a professor that you've taken a class with, or as it says here, you can go to the faculty websites and kind of get an idea of what all the different faculty members do for research, and find one that you find really interesting. Or another way to do this is, if you don't necessarily like or have an idea of what you like, find a professor that you really like and kind of get along with, because it kind of makes a big difference. So then as well, by going on the faculty websites, you'll have a list of their work and their contact information. So next step is to reach out to them. And so during these COVID times, it's a little hard to meet with them one-on-one. So you can reach out to them by email. If this wasn't during a global pandemic, I'd recommend to go find them in person and offer to meet them and have a conversation in person as well. So like a hint for this, to get in touch with these professors, is to start your email off by talking about some of their recent research. So I'd recommend going through and reading some of their recent publications and talking about specific papers that you found interesting. This kind of gives them a better idea that you're actually really interested in what they're doing, and you have an area that you found interesting, so it helps them kind of figure out a project for you. So when you're emailing them, be sure to include your name, your faculty, and your year of study, specifically why you're reaching out. So for example, a summer research inquiry. Mention that a few articles, as I mentioned before, that are published by them, and write what you found interesting, as well as why you're specifically interested in their research. And then lastly, you want to state any awards that you're eligible for and are interested in applying. This, again, my supervisor told me that this would really set, if you had a bunch of people emailing him, this would set certain people apart from the rest of the group. So it's a good thing to know what you're eligible for, what you're interested in, and that you intend to apply for funding. So now, once you have a supervisor, they'll help you through the application. There are usually, with these, there are a couple of not only, so there's student side of the application as well as professor side of the application. So chances are good you'll have to give a little bit of description of the project that you're working on. And as an undergrad, you're probably not going to know at all what that is, which is fine. I never knew what my projects were when I was really doing the application. My supervisor really helped me out. So having a good rapport and conversation about this, whether it be email or I guess Zoom now, it would really help. What I've done in the past is I'll write a quick little blurb and then send it over to my supervisor to edit, and then we kind of work collaboratively that way, based like me jotting down everything I know about what I'm doing and then he filling in the blanks about what it actually is. So the student deadline is usually about a week before the supervisor deadline. So also ensure that you are communicating the deadlines, because you can't really expect your supervisor to keep track of your deadlines as well as all of theirs, as there are times, like right now, there are a lot of grant applications going on. I know there's a lot of conference deadlines and abstracts are due. So they're super busy with deadlines. So it's easiest if you help communicate to them your specific deadlines. As well, one really important thing is, I know for sure the NSERC requires an official transcript. So make sure that you're ordering that in advance, and you can't just submit like one that'll come in a day. Usually you can pick it up in, I think it's McKinney, but I don't know if they're doing that right now. So ordering it ahead of time. If you get it shipped, I think it's taking about 2 to 3 weeks. So just planning in advance for these things. So one thing that's really important to note is oftentimes the supervisors are limited to 1 or 2 students per summer program. And I think this also applies during the year as well. So try to avoid overload on the professors and ask earlier rather than later. However, if they can't take you on, don't take it personal. It's probably not your fault. They are just have too many people going on or have too much going on. So talking to them like, oh, do you know anyone else in this area that would be interested or has space? Just trying to utilize that connection that you made with that person will help you kind of get ahead in finding a research supervisor. So, so what you can expect as an undergraduate researcher, once you get all your funding and all this, you'll usually start in May. So if it's your first time getting involved, or even if you've been doing it as a while as an undergrad, you'll likely be working really closely with either a postdoc, a graduate student, a research tech, or if you're in a small lab like me, you'll be working more one-on-one with your supervisor. So you won't be in charge of setting your own deadlines or even figuring out what you need to do next. They'll tell you what to do and then they'll give you deadlines and you have to kind of work on your own to meet the requirements and the deadlines. But usually it's pretty reasonable and they're there to support you through the entire thing. So if you get lost, don't really understand what's going on, they're there to help. And for me, one of the most exciting parts about being involved in research was being a part of a lab group. So for me, that means attending lab meetings every week and getting to know the grad students and the postdocs and everyone involved in the lab. So it's not just me being the lonely little undergrad in the lab working alone. There's a lot more to it, and it's really cool to kind of hear everyone who's an expert in their fields talking about it. So the chances are good at these lab meetings, you'll be discussing various things. My lab group does journal clubs all the time, as well as going over publications and upcoming presentations. So it's a really good opportunity to learn about the other side of research, which I'll talk about in a little bit, such as like what your professor is looking for in presentations or publications. And lastly, so again, you're the undergrad in the lab. It's not scary. They're not expecting you to be on a grad student level. So if you feel like you need support, feel free to reach out and talk to your supervisor. They're there to help you and they want to see you succeed. Guaranteed they wouldn't have taken you on if they didn't think that you could do it. So they're there to help you and be your number one ally in your journey in science. So tips. So my tips for being a new researcher is don't be afraid to ask any questions that you have, even if it's not related to your project, but you've read something and you kind of want to know more about it. Talk to your supervisor. They love to have these conversations as well. So I did a summer research project, but I also did a presentation at a provincial conference in the fall. So asking for more opportunities to learn about the field, or as the next point says, asking for opportunities to present. They kind of love that because that's the whole point of doing the research, is doing it, but then also disseminating that knowledge to other people. So you going to talk about and taking the initiative reflects really well on you as well. You want to take advantage of the one-on-one experience that you'll have with an expert in this field, whether that's talking to your supervisor or getting to know the other grad students and learning from them. I was able during my summer to help a couple of other grad students as well with their research. So for me, that one summer was really interdisciplinary and I got to work with tons of different approaches in the biomechanics field for me. So it was really cool and I just saw a lot of different things. So I got to know the field a little differently and saw how what I was reading in a paper was actually done in person beyond what I was doing. So one of the other big things is you want to get to know your fellow lab researchers or other undergraduates. 3 of my closest friends I met that first summer that I did undergraduate research, and one of my closest, like I go hiking a lot, so one of my closest hiking buddies was actually one of the PhDs in the lab as well. So they're people too, and they're really cool. So I would recommend getting to know them as well. It's important to remember that this is an opportunity to learn and to not be perfect. So I found so this was a new experience for me. So I did not have any clue what was going on often in the lab. So I made a lot of mistakes, and I learned pretty quickly that that was the job as an undergrad, was to make those mistakes and to learn from them. So don't be afraid to make them. That's kind of the whole point of this, and that's how we learn the most. So one of the big things as an undergrad, what I found the most intimidating was the presentations at the end of the summer. Often, some of the awards that you're available for are going to have a presentation requirement at the end of the summer. So it's not just you're doing this research and then you finish it. You actually have to go and talk about it and tell people what you found. So I have gotten a lot better at this. I was really bad. I would get so nervous before my presentations, but through practicing with the other undergraduates and giving the presentations to my lab, I got more confident. And just the key to do this, to overcome being nervous, is to practice as much as possible, so you really know what you're talking about. So as I mentioned before, the lab meetings are a great opportunity to practice and receive feedback. So you'll really have a nice polished presentation before you get going. And if you're unsure about any part of the presentation, talk to your supervisor and they'll have some great tips to get you through. So lastly, we are in a global pandemic, so research looks a little bit different during the time of COVID-19, but you're still able to do it. I am doing my second project right now from home, and it's not really any different. I just don't see my supervisor as often, but he's still available via Zoom. So if the global pandemic is a limiting factor, you're not going to get the same experience. Don't let it limit you from getting involved in research. Your supervisor will still have projects for you to do. Science hasn't stopped. In fact, like, there are so many new opportunities than there ever were before. So with this, we have a lovely list of awesome profs who have offered their time and volunteered their time this evening to talk. So we'll get started with that. I will stop sharing my screen, and then I think we have first Dr. Steve Vimosi. And if I mispronounce your name, I apologize. So Dr. Steve Vimosi is a professor of population biology in the Department of Biological Sciences, scientific director of the Kananaskis Field Stations, and associate dean, equity, diversity, and inclusion in the Faculty of Science. He is originally from Ontario and has been in Calgary since 2003. His broad research area is evolutionary and conservation ecology, with a special interest in threatened fish and amphibians in the Northern Hemisphere. So with that, Steve, do you want to take it away?
All right. Well, thank you so much. Thank you so much for the intro. And yeah, just thank you so much for the night. And that was a really fantastic presentation. And if you saw me pop it in the chat, but I mean, Dave Eaton had a really good point, really good advice. And I think certainly about the email piece, I see a lot of prospective grad students who seem not to know about your advice. So it's good. Keep sharing that. So again, as you said, I'm Steve Vimosi. I run a conservation and evolutionary ecology lab. So right now it's a group of 5 of us. So of course, me as the PI, postdoc, Christy, she is in the foreground here in this picture that I'm going to keep using as often as I can. Emily, they're doing a PhD, and Charity and Jared are doing masters. Charity's hopefully finishing up soon, and Jared just started. The beasts that we're working on, or will soon be working on, mostly aquatic, semi-aquatics, so bull trout, Chinook salmon, and long-toed salamanders. I knew we didn't have a lot of time, so I'm just going to kind of jump into the 2 projects that'll be happening this summer, where there's active research going on, and we're looking for folks. So the first one is some continued work on bull trout. So this is actually a picture taken of a former grad student, Emma, holding a lovely fish here. And really, in this case, it's we're looking, so again, bull trout, it's Alberta's provincial fish, and unfortunately threatened. Lots of things happening. So climate change, warming waters, sediment in water, and fishing, and introductions of non-native species. So we're really trying to get a sense of, where are they still doing well? How are they doing at the range margins, which unfortunately keep kind of going up mountains, so they're shrinking. And then also a rather specific question. There's some really nice work being done by the government, and in terms of trying to remediate portions of the Tay River. And so working with Alberta Environment and Parks biologists, we're trying to ask whether or not those remediation efforts are actually doing anything positive. So the approach is, obviously in both cases, I didn't write it, but field work. So you would get to go out in the field, doing some really nice stuff with environmental DNA. So, assessing whether they're there by some of the DNA products left in the environment. So you're not having to catch and handle a lot of the fish. And then also looking at the environmental variables at those sites. So where that would take you, hopefully, we will get to do some field work in a distance way. And that'll be work on the east slopes of the Rockies from Kansas country up to about Caroline, but also Christy wants to go, and she's had some really good interactions with some good dialogue with some First Nations communities up in the Yukon. So, and that was set for last summer, and of course, but the Yukon wisely shut its borders, and so that obviously was paused. We're hoping that happens this year. That's the first one where we're looking for a student. And then the second one, I think it's right on top. So the second one is looking at basically Chinook salmon. So some of you might know, I mean, most of us like to eat it. I certainly do, but they're threatened again. And we're really interested in populations living in the Yukon River. And especially we've been in, again, dialogues with the Tlingit from Teslin. And their issue there is they're, hundreds, if not some like 1,000 kilometers inland. They're one of the most furthest inland sites where salmon get to. And these salmon first have to go from international waters into the US before they get into Canada, before they get to them. And so unfortunately for many years now, they're basically not fishing. And of course, this is a economically and culturally important species for them. I think a couple of years ago, they let themselves catch 3. And that's just not anywhere near what they would have been able to do in the past. So here, the question, a really nice project that master's student Jared is looking at is basically trying to reconstruct some of the historical abundances using tree coring. So basically, as these fish die, they fertilize the ground. And so you can actually pick up in the growth rings evidence, and you can kind of correlate that with rough abundances. So we want to, again, work with the Teslin community to kind of come up with a different, they're of course going to have good, rich story and rich oral history on this, but working with them to introduce a different, give them more data essentially to make the case for protection and stuff like that. Same thing, going in the field, this time tree coring, so whispering to trees, hoping the trees whisper back, some site assessments, and again, working in south central Yukon in this case, almost exclusively. All right, and then so those are the 2. Oh no. And so basically, again, as I mentioned, we're recruiting at least, but probably just the 1 undergraduate student for each of the 2 study systems. And there's my name again, and there's my email. So thank you so much.
Awesome. Thank you, Steve. Your research sounds very interesting. I might have to go look up some stuff on salmon now. So next up, we have Dr. Jessica Theodore. She is originally from Toronto and has completed her BSc in paleontology at the University of Toronto and her PhD at the University of California, Berkeley, and has been a professor at U of C since 2006. She's a paleontologist and evolutionary biologist studying hoofed mammals. She studies their morphological evolution, adaptation to the spread of grasslands, and the evolution of large body size in mammals. Her fieldwork is primarily in the Cypress Hills of Saskatchewan, but also includes some areas of southern Alberta. If you want to take that away, Jessica.
Sure. Okay. Get some slides up. My slides are not nearly as fancy as Steve's, but hopefully you will enjoy seeing some of these things. So I'm a paleontologist. I study extinct hoofed mammals, and my interest is really understanding herbivore communities and how they've changed over time. both in terms of how they've adapted to grasses and changed what they eat, and how they interact with each other. So this is a scene from a reconstruction of what would have been around in North America 60 million years ago. So after the extinction of the big dinosaurs, but before anything that you would consider a modern fauna. And then this is about 20 million years ago, and you can see it looks a little bit more normal compared to what you see here. There's a lot fewer things here that look anything like anything alive today. And it includes things like primates, which are no longer certainly in North America. So my fieldwork is mostly in the Cypress Hills in Saskatchewan. We haven't done much in the pandemic, but we also do have some localities in Southern Alberta that are a little easier access and don't require going across provinces and dealing with all sorts of paperwork right now. A lot of the work we do involves looking at morphology of fossils. So some of the work we do involves sorting through little tiny teeth that we've collected. We gather a lot of, when we find larger fossils, we also gather dirt and we sort through it. We screen it and we sort through it under a microscope, looking for tiny teeth to identify what else is there and get an understanding of the site. But I also have a number of students who have done work over the years creating 3D models from CT scan data so that we can understand the evolution of these things by looking at the evolution of their skulls, and particularly the inside of the skulls that we can't access otherwise without cutting up skulls. And so I have currently got 4 graduate students, 2 PhD students, and 2 master's students. One of the PhD students is actually working on dinosaurs, and the other is working on camels. You may not have realized it, but camels are actually originally native to North America, and most of their evolutionary history is here, not in Asia or South America. And this is just a sampling of some of the camels that have been around through time. Everything from little tiny camels, this is, you can see relative to a human silhouette, to one that could look through a 2nd-story window. And that brings up kind of my last sort of research interest, which is the evolution of large size in mammals and why things like camels never got to be as big as dinosaurs. And I think I'll probably stop it there and pass along to our next speaker. And if anybody's interested in doing any of this work, this is one of my former undergrads in the field. Michael's now an environmental consultant, and that's my email address, and you're more than welcome to contact me. Thanks.
Perfect. Thank you so much. That also sounds so interesting. All right. So next up, we have Dr. David Eaton. He was appointed from 2015 to 2020 as the NSERC Chevron Industrial Research Chair in Microseismic System Dynamics in the Department of Geoscience. With graduate students and postdoctoral fellows, his work focuses on advancements of research education and technological innovations in microseismic methods and their practical application for resource development, with a secondary focus on enhanced graduate training in STEM disciplines. His work has been recognized by a 2019 NSERC Synergy Prize for Innovation and the 2020 J. Tuzzo Wilson Medal from the Canadian Geophysics Union. So with that, want to take it away?
Great. Well, thank you for that introduction and this opportunity to be here. You'll be pleased to know that most of what you talked about there, I'm not going to be talking about tonight. My background is in seismology. I'm a professor in the Department of Geoscience, and I study earthquakes, and particularly earthquakes that are caused by humans. That's been a really interesting topic for me to look at for the last 6 or 8 years. But what I'm going to talk about tonight are 2 side gigs that I have going on, 2 projects. The first one here is called Redevelop, which is a responsible development project. It is a project that involves 5 universities altogether. It's a training for graduate students in multidisciplinary teams, so science, engineering, public policy, and business. And what we do is engage with leaders from lots of different areas, notably Indigenous communities, but also industry and government, to address important problems. And you see the tagline is training Canada's future leaders in low carbon energy. And that's really what it's all about. And as we all know, the energy sector is changing very rapidly right now. Now, as part of this program, we hire Indigenous summer undergraduate interns. The last time we did that was 2019. We skipped this year because of COVID, but we're planning to do it again, virtual internships in 2020 or 2021. So if that is of interest to anyone, these are the two Indigenous interns that we hired as part of Redevelop in 2019. They got placements in science labs. The kind of work they did actually was quite a bit like what Becca was describing earlier. They were assigned mentors. They were given real projects. They presented their work at the end. It includes people from elsewhere. So Joseph Ledley is from Mount Royal University, and he was really interested in water quality issues. And here he's doing some sampling. So it's a chance to develop analytical, critical thinking, and communication skills. So if you're interested, here's the person you should contact, Celia Kennedy. So she's the program manager for this Redevelop program, and you can see her email right there. So that was the first project I wanted to tell you about tonight. The second one is a new project that just got spun up during COVID this year. We call the project NIAC, which is a Cree word that means for the future, but it's also an acronym. It stands for Network for Indigenous Youth Academic Knowledge. So this one is funded by NSERC's Promo Science program, which is a program which is all about promoting science to youth. What we're trying to do here is to introduce Indigenous students to undergraduate and graduate degrees in STEM disciplines. For this, we're working really closely with the Sucker Creek First Nation, which is on the shore of Lesser Slave Lake. And they're really interested in water quality issues. So they've noted changes over time in ecosystems and the water quality. The youth in this community are interested in trying to work together to do something about that. And so this program is leveraging that interest to try to encourage more generally students to go into to STEM disciplines. So this has been a great program that we just started working in. You can see part of the philosophy here. What we're trying to do in this NIAC program is to mix together Western disciplines and the natural sciences along with traditional knowledge. Both this as well as Redevelop, this is to feed into informed decision-making. So again, if anyone has questions about either of these two programs, feel free to reach out. You can see my email listed there. The website for the Redevelop program is redevelop.ca. So thanks a lot. That's my presentation.
Great, thank you so much. Okay, next up we have Dr. Sean Rogers. He is a professor of biological sciences and the director of the Banfield Marine Sciences Center, U of C's Oceanside campus. Sean's group focuses on ecological and genomic approaches to understanding the consequences of environmental change for species, especially towards the conservation of coastal fishes. All right, take it away.
Great. Thanks for that, Becca. And it's really good to be here with you all tonight. My name's Sean Rogers. I'm the director of the Banfield Marine Sciences Center. We call this your Oceanside campus because U of C is a co-owner of this amazing facility. It's located on the west coast of Vancouver Island. In a normal year, we would have over 100 researchers, both national and international, out at the center. We typically offer about 18 courses a year, field courses, undergraduate field courses, and we have approximately 50 staff and around 3,000 students annually visit. It's a little quieter this year. We still have our 25 core staff and researchers that have been relying on us staying operational for critical research. So we still do have researchers working out there and keeping the place operational. So our research in the Rogers Lab is focused on ecology and evolution. A lot of our current research is focused on questions surrounding how organisms cope with environmental change. We currently have an NSERC strategic research project in partnership with Hawaiian First Nations and the Dididat First Nations on coho salmon. And this project, up on your left, you see a picture of the Nitnat River, which goes into a saltwater lake, Nitnat Lake. And we're really interested in understanding the migration of Pacific salmon, and specifically, what helps them survive the journey out to the ocean and back into the natal rivers in which they spawn. So we take a multitude of approaches, everything from studying behavior and physiology, biomechanics, to going back to the lab and studying genomics and molecular ecology, DNA fingerprinting. Right now, our project is trying to understand, we've noticed, and the First Nations had noticed out there, that when you give salmon toys to play with in hatcheries at a young age, more of them actually survive to come back. And so this can be anything like twigs and cover in a tank to other types of enrichment. So we've actually been testing this experimentally. This is an experimental setup that my student Alex just built to study incubation and early life stage survival. And yeah, we track the salmon from the time they leave the hatchery to the time that they get back. So we are actively engaging undergraduate students for these projects. There is a fairly good chance that you would be able to do field research. Of course, it's all dependent on COVID, but we typically really aim to give our students an authentic research experience. And in some cases, students choose to live out at the BMSC for the duration of their research, which we also support. So, so with that in mind, we also have a number of other projects on coastal fishes that I don't talk about here, but you can certainly reach out to me, and you can check our website in terms of some of the ongoing projects happening at the BMSC right now. And I've included a picture of my lab. This is what our current lab meetings look like, which I suspect is much like other lab meetings in this time and age. So with that, please do feel free to reach out at any time, and I'd be happy to answer more questions about the opportunities there for other researchers too. We have a number of researchers in Banfield, so if you're interested in knowing about other opportunities, just let me know. Thanks, Becca. I think that was 3 minutes.
Pretty close. All right, thank you so much, Sean. That sounds so cool. I'm kind of jealous that your undergrads get to go stay at the BMSC, but that's okay. We all choose our research areas, right? All right, so next up we have Dr. Belinda Hain. She is a professor of chemistry, and her research is based on the use of light to solve society grand challenges. So you want to take it away, Belinda?
Thank you. I'm going to try to share my screen. Can you see my screen?
Yes, we can.
Yes, perfect. Okay, so I'm a professor in the chemistry department, and we heard a lot about like very cool project in biology. So I hope I will be able to do something as cool as that. So my research is very interdisciplinary. We are really working at the frontier of everything and you're going to see from biology to chemistry to physics. So there is not one single thing that we are not doing in the lab, and very often MacGyver is becoming a verb in my lab, and we are MacGyvering things. So really what we are trying to do is use light and simple light bulb in order to solve society's grand challenges. And the grand challenge that we are the most interested with is really the one of antimicrobial resistance. So we see there is a biological application there. And as you might be aware, a failure to address antimicrobial resistance, although we are living in a pandemic, the world seems to be even like worse in the future, as if we are not doing anything, about 10 million of people are going to die every year from a simple infection. So that means that a simple paper cut could lead to your death by 2050. 10 million of people dying from an infection is more people dying from an infection than cancer and diabetes right now. So it's going to, in addition, cost enormously to a society. It is estimated by 2050, we are going to spend 100 trillions a year just to combat infection. And so I have really dedicated my research in trying to find alternative way to combat antimicrobial resistance. And one of the ways is photodynamic therapy. So we are using light. We are simply using the oxygen present in the atmosphere, and we use a dye, a photosensitizer, in order to simply give the energy of the light to the oxygen. And what we are doing is really, we are taking something which is as nice and inoffensive as Mickey Mouse, and we're providing it energy, and this is what is ending up. So we are creating something which is extremely reactive, and that can react with bacteria, both gram-positive, gram-negative, but as well go up to the fungi. Compared to the antibiotic, we have a much broader range of capabilities. And very recently, we have been called upon to work with the World Health Organization, CDC and NIOSH, to work on COVID. And we have demonstrated that we can disinfect, basically for reuse, mask, so the PPE, so the mask that the people are using in the healthcare system when we had such a big shortage, so that like people could use more than one time, things that you shouldn't be able to use more than one time. And so here is the publication that we have submitted in the medRxiv, and we are submitting it right now for review at a very big journal. It's basically, we have been working, we are like, it's across many continents, and as well, like many people, we have 49 people here. What's next? I'm almost done. We're trying to basically go from production of single oxygen, which is that small, to something which is that big. And we're using nanotechnology often in my lab. So I have a research group composed of like one PhD student currently. I have 4 graduate students, and I have had many undergraduate students over the years. Some of them in all of those pictures. We are trying to have, as you can see here, a lot of fun. We are going and doing quite a lot of activities. The research is funded by NSERC, the University of Calgary, and you have the link to my website, my Twitter, and as well my email address if you wanted to contact me. We are always welcoming undergraduate students to come and do some research with us. And I'm at 4 minutes 15, I'm sorry.
All good, thank you so much. I just wanted to see, because I didn't see her name, but is Dr. Kathy Ryan in the call? I don't see her. Okay, so next up we have Dr. Dan Sugar. He is a professor in the Department of Geoscience and director of the Environmental Science program. He is a geomorphologist, meaning that he's interested in how landscapes evolve, and he studies environments ranging from seafloor to mountaintop. Most of his work takes place in the high mountains of Northwest North America, though he uses a lot of satellite data from all over the world. If you want to take it away, Dan.
Thank you. If I can find my PowerPoint here. Oh my God, where did it go? Now I'm like that guy. All right, hold on. Oh, I had it a second ago. There it is. Okay, and now where's the share screen gone? Okay, you can see my slides. Yeah.
Yes, you're good.
All right, great. Thanks for that. So my name is Dan Sugar. I'm a prof in geoscience, and my lab's name is Watershed Lab, which is an acronym for Water, Sediment, Hazards, and Earth Surface Dynamics. But it's also, well, it's just that. We study sort of geological processes, relatively short-lived or modern geological processes across entire watersheds. So sort of from mountaintop to ocean floor. My email is at the bottom there or towards the bottom. That'll be on all the screens if you want to get in touch with me. So as Becca said there, I work on a variety of types of environments using a bunch of different kinds of technologies. You might have seen in the news a month or so ago, there was a gigantic landslide in or near Butte Inlet in coastal British Columbia that triggered a tsunami that was 100 meters high. And this event or series of events, sort of a domino of a cascade of events, if you will, is the kind of thing that gets me excited scientifically. And so I'm working with a few dozen other folks, geologists, but also ecologists, oceanographers, and the Hamalco First Nation to understand this, this pretty rare event. And I say rare because it was very large, but it was also a whole series of geological processes that that occurred in kind of a domino fashion. So on the right-hand side there, you can see a photograph from the air, and what happened was a landslide, a very large landslide, was released more or less from where the photograph is and went into that lake. In fact, you can see a bunch of debris in the lake there, which is some of the landslide material. And it triggered a tsunami that was about 100 meters high that overtopped the dam at the end of the lake and formed a big flood down the valley, which eventually reached the ocean at Butte Inlet and then formed what we call a turbidity current that flowed 60 or 70 kilometers along the ocean floor. So this is my current lab. Holly on the left here and Simon on the lower left are both undergrads working, doing satellite remote sensing work right now because we can't do any field work. Megan and Griffin here are both master's students, also doing remote sensing work because we can't do field work. And Mike is a postdoc who's just about to start in about a week or so. And so what I wanted to show you was some satellite imagery of two projects, one that's ongoing and one that's that we're going to start soon. So maybe on the lower left there, you see is sort of an animated GIF of a coastal area in Southeast Alaska. This is in Glacier Bay National Park. And this river here, which flows from the top and enters this lake and then goes out to the ocean. This is the Alsek River. And you can see that the glacier here is that it actually separates these two lakes. And between the two images, which are from, I think, 1984 or so and 2018, something like that, you can see the glacier is retreating quite a bit. And so what we're interested in is understanding whether in the coming decade or 2 decades, whether this glacier will be gone and these two lakes connected. And if that happens, whether Alsek River might enter the ocean here instead of here. And this is about 20 kilometers. And that might not seem like that big of a deal, but several species of salmon migrate up the Alsek River through Southeast Alaska into British Columbia and then eventually into the Yukon. And so the Champagne and Aishak First Nations in southwest Yukon are really concerned about the potential that salmon won't be able to migrate up this system anymore because of the cultural importance of those fish. On the right, upper right there, it's a satellite image of a lake in coastal BC called the Wichino Lake that is right by the ocean. The ocean would be just sort of at the edge of the image here. And each of these little red dots or red polygons is an individual landslide. And my undergrads, so this is an undergrad project where Simon has mapped all of the landslides. He wrote some code to map all of these landslides automatically using the Landsat satellite records over the past 30 or 40 years. And we're doing this work to basically understand what the risks are of landslides in this basin. And we're hoping, I've got my other undergrad, Holly, to do a similar kind of analysis in Butte Inlet, where that giant landslide I showed you at the beginning was. And so what I'm showing you here are two projects that, I've don't necessarily need people to work on these exact projects, but these are the kinds of things that I work on. So if you're interested in really rapid changes on the Earth's surface, if you have any kind of background in GIS, definitely come talk to me, because there's always projects to work on. And with that, I'll turn it back over.
Awesome. Thank you so much. All right. So next up, we have Dr. Mark Poulin. He is with the Hotchkiss Brain Institute. So Mark, if you want to take that away.
Okay. Thank you, Becca. So can you see my screen okay?
You bet.
Okay. So thank you very much. And first, I'd like to congratulate you and your team for putting this on this evening. I've been here for 20 years at UFC, and this is one of the most innovative things that I've seen. It's summer students are really the engine of many labs, including ours, and it's great to see this amount of effort and also interest from the students. I would say that I'm a member of kinesiology, but also the Faculty of Medicine, so cross-appointed in 2 faculties. And I won't be presenting tonight, much about my own work. I want to talk a bit about the Brain Create program. This is a interdisciplinary training program for new neurotechnologies. It spans a number of different faculties across campus, and we have dedicated funding for summer students, including Indigenous students, for the next 5 years. The program is really brings together a number of different physiological systems. It's personalized for the students' needs. A lot of these things here, you'll see it's really for graduate students, but a lot of it is applicable for undergraduate students too. There's an element of mobility, mentorship, and really we're focusing on this concept of extreme physiology as a testbed for innovation. So extreme physiology could be in terms of environmental conditions, it could be geographical conditions, but it also can be biological extremes. What we've done is we have a team of many scientists that have a cross-cutting themes in the areas of cognition and behavior, neural imaging and monitoring. This would be mostly neuroimaging techniques, model systems. This would be animal models, neuromanipulation and modulation. This is related to clinical populations, in particular populations with spinal cord injuries. Data science, heavy on computer science and machine learning and artificial intelligence, and then integrated physiology, which is brain, heart, and lung physiology. And we have a number of industrial partners on board, and we essentially try and bring the students together with some of these different types of experiences to focus on a project of choice. So if there's anyone interested, I would encourage you to maybe reach out, and then we can connect you with the people in your areas of interest. In particular tonight, what I wanted to just share is the mobility component of the program. So we have opportunities for national and international mobilities, although, as everyone knows with COVID, some of the things are on hold. It's unclear how things are going to work out this year, but going forward, we expect to be back on track. In particular, our mobility components bring us to the Canadian Arctic and to remote parts of the country. In the Arctic, we've established collaborators and partners in different parts of the Arctic, including the Yukon and the Northwest Territories and Nunavut. We've done some work at the Kluane Lake Research Station. And another part of the component, which is really part of the living laboratory research experiences, is work we've done now for a number of years in northern Chile. This shows you some of the scenes here from the moraines and the glaciers in the Yukon. So why the remote environments, in particular the Arctic? Well, there's a number of things that come together in these unique environments, including some of the challenges related to physiological, psychological, and cognitive interactions of being in remote environments, and some of the adjustments that might take place with either, longer darkness during the days in the winter and more sunlight in the summer. There's also opportunities for remote sensing and telehealth, and learning about and also building research bridges with our remote community, northern community partners. This slide here is a slide that really highlights our work in Chile at the ALMA observatories. This is one of the world's highest observatories. There's a huge proportion of the population in Chile that works at high altitude because of the geographical nature of the country, as well as the resources in the mountains. This here shows you a scene at about 2,000 meters above sea level. This is the base camp where several hundred workers will be positioned here to work 1 or 2 weeks at a time. And every day during those 1 or 2 weeks, they take a 45-minute drive with SUVs and so on to this place right here, which is at 5,500 meters above sea level, where you will find about half the oxygen that makes it to our body compared to what we have at sea level. And so what we're really interested in is to look at the effects of this sort of extreme environment, so low oxygen, on the way the body functions and the difficult tasks that these workers undertake. The goal for this program was to bring a number of students to this environment every summer for the next 6 years. And I must say that we're not necessarily focusing just on physiology. I've heard a number of really interesting projects here that potentially could apply to these sorts of remote environments. So we're also interested in partnering with some of the other researchers that have presented this evening. And what I wanted to do is just show you some pretty fascinating images from the skies in northern Chile. This is, as I said, one of the world's highest observatories, and it's the world's largest millimeter microarray to look at the night skies. I had actually a number of additional slides, but it looks like they didn't overlap properly. Maybe it's with the screen sharing, but there's a number of difficult tasks that workers will take up at 5,500 meters, including some of the work on some of the most powerful computers in the world. And so, errors in decision making or in processing information that due to low oxygen can lead to multi-million-dollar errors and a lot of downtime. So we're trying to understand the physiology and the cognitive effects of this low oxygen on the decisions that these workers make, and also on the acclimatization of these workers to these high extreme environments. So that's all for me, and happy to turn it over to you for the rest of your program.
So thank you everyone for coming. We are at the end of our time. For those who would like to stay and participate in breakout rooms, I will make them. But if you're a professor, please know that you don't have to stay to the end. You've all left your email addresses, and if there's something you'd like us to share with students on your behalf, you can. I'm gonna really quickly make the breakout rooms, but if you'd like to leave, feel free. Thank you so much everyone for coming. I don't want to keep anyone longer.
Thanks everyone. Good night.
Good night. All right.
Take care.
Thanks so much.
I'm good to stay for a bit.
Okay, so I think we have 4 professors. Dr. Jennifer Adams is also here, I believe, and she didn't have the chance to chat. But I'm just gonna make the 4 breakout rooms, and if you'd like to head to one of those rooms, you're welcome to. And if not, that's okay too. So there's 4 rooms. You should have the option to choose. I'm just gonna assign the professors to a room. So Dan is in room 1. Jennifer is in room 2. Mark is in room 3. Steve is in room 4. And oh, Jessica needs a room as well. Is it all right, Jessica, if we use this room for you? I don't know how to open another one now. Is that okay?
Yeah.
Okay, so everyone just head to whatever room you'd like. Um,
Does anybody have any questions they want to ask?
I actually have a question for you, if you don't mind.
Of course I don't mind.
I'm in engineering, so it's maybe a little bit different. But I was just wondering, what qualities do you think like make a student well suited to research, or like for students who are thinking about research, what do you think would be some considerations?
So the thing that I usually do when students start in my lab